So our next speaker, are you here? It's yes, Aleksandar Jeremic.
Yes.
We are staying in the neighborhood of diabetes, but moving from computational methods to experimental ones. Aleksandar is from George Washington University, studying the roles of copper in the aggregation and toxicity of human islet amyloid polypeptide. Aleksandar, please.
Thank you, Yochid, and I would like to thank Rams for this nice invitation to participate in this very interesting symposium on the role of metals in aggregation toxicity of amyloid proteins, which is actually the title of my talk. We have a long-standing interest actually in role of amylin in relation to diabetes, and I hope that my presentation will provide some answers on that question that someone asked about the role of amylin in diabetes. But before move on and discuss with you the role of amylin in diabetes, I would like just to give you some introduction on how amylin may contribute to diabetes for those of you who are less acquainted with amylin biology. Diabetes is a multifactorial metabolic progressive disease. It affects worldwide populations. Just alone in US, it's accounted that about 1/3 of population will be directly or indirectly affected by diabetes. And one of the key attributes of diabetes is actually dysfunction of beta cells that cannot supply our tissues with enough insulin, which is the main glucose, together with amylin, main glucose regulator. As we all know, type 2 diabetes mellitus is a multifactorial disease. Genetic, environmental factors, as well biochemical metabolic events leads to type 2 diabetes mellitus, including insulin sensitivity is one of the confounding factors, beta cell demise induced by ER or redox stress, islet inflammation, and the title of my talk today, and actually long-standing interest of our lab, is to understand the role of amyloid formation, islet amyloid, in an amylin toxicity in progression of diabetes. Many factors may contribute to amylin aggregation toxicity. Some of these pathways are summarized in slide that is in front of you, including redox pathways, degradation pathways, and other trafficking recycling pathways. They may regulate amylin turnover and amylin toxicity in beta cells. As a colleague of mine just remind you of the fact that insulin may contribute to islet amyloidosis. Interestingly, amylin is the main confounding factor in, at least in, islet amyloidosis, and contributes to progression of diabetes and destruction actually of beta cells. On the right side of this slide are the two post-mortem histology slides of patients that died from diabetes, and actually they all harbor, over 90% of the patients harbor amyloid aggregates, form amyloid aggregates in their pancreas. Biochemistry and biochemical evidence suggests that the reason for aggregation of amylin, human amylin, is this amyloidogenic segment between 18 and 28 amino acids that contains several important amino acids that may contribute to interactions with many factors, including metals. And today I will try to concentrate on this part of my talk, discussing these amyloid segments and how may interact with the metals to modulate amylin aggregation and amylin toxicity. Interestingly enough, rodents that has these three proline substitutes, amino acids, which are known as beta sheet breakers, never actually harbor and never produce amyloidogenic forms, in contrast to human counterparts. What is known so far, what we know about the mechanisms of amylin toxicity, well, it progresses and follows more or less same biochemical, biophysical mechanism of aggregation that is shared among many amylogenic proteins. Starts with assembly of soluble oligomers, then proceed toward insoluble beta sheet enriched fibrils. However, recently another hypothesis gained popularity that these off-pathway so-called soluble small toxic oligomers actually are predominant toxic species. One way or another, whether it's oligomeric or more mature aggregative form of amylin, is actually main culprit in isolated amyloidosis and beta cell toxicity, is something that will be a matter of discussion in the future as well. But today I would like to focus more on the factors that regulates oligomerization and aggregation of amylin. Fact that under normal physiological conditions, isolated amyloid and amylin-derived aggregates are rarely seen, suggests there are obviously confounding factors in the pancreas that stimulate or maybe even inhibits aggregation of amylin. High-resolution images, such as this AFM image of amylin, clearly shows a very regular pattern of formation of these amylin fibrils formed from this linearly arranged smaller oligomeric species, or that has very small dimension of height of about 6 nanometers and diameter of only 90 nanometers, that align in this bidirectional fashion to form elongated fibrils. Just before I go and start discussing the main topic of my talk today, that's a role of copper in modulation of amylin aggregation and toxic properties, I would like just to point to you the importance actually of knowing the factors that control amylin aggregation. Because transgenic model system, such as this transgenic mouse that overexpress human amylins, tend to produce these very toxic plaques, illustrated here with asterisks. These purple regions within the individual islets represents amylin-derived amyloid plaques that tends to take the space and destroy islets, and including beta cells, and in this progressive manner, so that after a few months, these amylin aggregates will eventually lead to full destruction of islets, leaving just exocrine tissues without ability of these mice to regulate glucose levels, and consequently, the glucose levels go from normal to very high levels, basically evoking type 2 diabetes condition. So obviously, knowing the factors that control amylin aggregation, factors that may stimulate or factors that may inhibit amylin aggregation is of big importance. And recently, metals such as copper and zinc obviously become one of the prime targets for as modulators of amylin aggregation and toxic properties, which was also a matter of our interest, our focus in our lab, and rationale for our study exploring the role of copper in amylin aggregation, amylin toxicity, and pro-oxidative ability of amylin to generate reactive oxygen species, came from relevant studies done in with other amyloid type of proteins, particularly beta amyloid, that show that in the presence of copper, they can presence of copper stimulate ROS formation and there is apparently synergistic effect between copper and beta amyloid and other proteins to produce reactive oxygen species. Interestingly enough, copper levels are both elevated in the pancreas and as well in serum. And this actually led us to propose a hypothesis, and with an analogy with similar studies done in related amyloid diseases, that copper and human amylin may act actually synergistically to produce more ROS and to reduce oxidative stress. And in other words, that copper may mimic its effect on pathological action of other amyloid proteins. Here, and I would like to address two important biological and pathological relevant questions. First, does fibrillation process itself cause ROS formation and accumulation, at least in vitro? And is there any causal relationship between copper and amylin catalyzed generation of ROS and apoptosis of pancreatic beta cells, which are two primary features of type 2 diabetes mellitus? So first, we embark on studying amylin aggregation and oxidative properties in the absence of copper, just to establish the baseline for our studies, and then compare in the presence of copper. On the left side, you can see that amylin, and that was actually previously reported in the literature, that amylin and both human amylin and rat amylin can produce minuscule nanomolar concentration of hydrogen peroxide, which we found out later that is not related to its aggregation properties. Why? Because when we use catalase to completely inhibit amylin induced hydrogen peroxide production, we didn't see any change in amylin aggregation properties examined by THT fluorescence. Of note, rat amylin, non-amylogenic form of amylin, also produce similar or comparable amounts of hydrogen peroxide comparing to aggregation from human amylin, suggesting there is really no correlation, and some studies suggested between amylin and amylin-induced ROS production and amylin aggregation. Further studies actually confirmed, studies confirmed that addition of hydrogen peroxide on itself did not modulate any aggregation properties amylin. Increasing concentration of hydrogen peroxide really did not change much THT fluorescent values. What we found, interestingly, that amylin, and that both amylin and rat forms, so both isoforms of amylin, were capable of both producing and quenching hydrogen hydrogen peroxide, which is illustrated within the figures B and C. So exogenous addition of hydrogen peroxide was reduced in the presence of both human and rat amylin, and figure C here on the right side illustrates basically these quenching properties of human and rat amylin. Having established that that amylin can produce this relatively modest amount of hydrogen peroxide, we wanted actually to extend the studies and ask whether copper in some way may, based on a hypothesis, may induce or may stimulate this hydrogen ROS production. But before we could answer these questions, we wanted to actually to assess effect of copper on amylin. Basically, we constructed amylin copper binding curves using tyrosine fluorescence as a proxy for amylin interactions of copper with amylin, and these binding curves demonstrated, and this is here represented in C and D, demonstrated formation of low affinity complex in the micromolar range, which is actually in good agreement with literature and other amylogenic protein, showing stoichiometry of 1-to-1 binding between copper and amylin. What we didn't expect to find, and in contrast to our hypothesis, actually was that copper, rather than accelerating amylin aggregation, copper had inhibitory effect on amylin aggregation, basically cut in half the amount of amylin aggregates in the presence of copper, and slowing down kinetics of amylin aggregation, slow down basically the rate of amylin aggregation by almost half. Kinetic studies reveal that copper inhibits amylin aggregation by slowing down, actually working mostly on kinetic part of amylin fibrillization, reducing energy of activation without really affecting the mechanism of amylin aggregation, which is shown here in this slide B, showing exactly the first rate order of reactions in the presence and absence of copper. Then we went on and trying to explain the reason for this inhibitory effect of copper on amylin aggregation. Amylin, like other amyloid proteins, aggregates in very organized manner, going from monomers into the oligomers and then beta sheet enriched fibrils, and this can be examined using structural techniques such as CD spectroscopy that examines secondary conformations. So in front of you are these time-dependent CD spectroscopy studies of human amylin in the absence of copper and B in the presence of copper. And one can see increased progression from random coil monomeric structures into the beta sheet enriched fibrillar structures, which is actually probably even better seen on these lower slides, showing a minimum of 224 nanometers, which is spectroscopic signatures of beta sheet enriched fibrils. Again, in the absence of copper, when in the presence of copper, both at 4 hours and even after 48 hours, we see amylin preserving its native soluble, predominantly random coil structure. So this structural studies explains basically the inhibitory effect of copper on amylin aggregation observed with theoflavin, the assays. Then we move on to, in addition to exploring the effect of modular effect of copper on aggregation of amylin, we also examine the effect of ROS production, and for that we use a combination of copper reducing reagents such as ascorbate and glutathione, which will reduce to copper 1 and produce, as we already heard today, significant amounts of hydrogen peroxide and hydroxy radical shown here in slide B. We just established here the baseline for ROS production catalyzed by copper. Interestingly, in the presence of either human or rat amylin, but particularly human amylin, there was no further potentiation of ROS production by copper in the system, as some studies suggested that it may happen. Actually, we found out that presence of amylin, not only that we did not enhance copper catalyze ROS formation in vitro, but also in some instances, such as, for example, in presence of ascorbate and copper, there was a significant decrease in production of ROS, both hydrogen peroxide and hydroxy radicals. If you compare this versus here, one can see there was a significant decrease in ROS formation in vitro in the presence of amylin. This was confirmed in rinse cells, so using in cellular conditions. So when we challenge ourselves with copper and human amylin, there was a significant actually decrease in pro-oxidant effect of copper and human amylin when we challenge ourselves as a copper human amylin complex in agreement with this in vitro studies. Then we follow up with the classical cytotoxic studies, basically confirming that using either MTT studies, which is a proxy for metabolic state of the cells, and also indicative of toxicity or, if you like, viability of cells, showing the kind of similar finding in the presence of human amylin, in presence of copper, human amylin toxicity was significantly less. This was reconfirmed with Western blot study showing less stress kinase activation of these pro-apoptotic kinases pathways were less in presence of copper versus absence of copper. And finally, truly apoptotic assays such as using caspase-3 and Nexin staining showed human amylin toxicity reflected by increase in caspase-3 and Nexin staining, and significant reduction in amylin toxicity in the presence of copper, which was interesting finding and in opposition with our starting hypothesis. Previously, it was suggested that metal chelating agents, such as, for example, batracopene, may be helpful against the amylin toxicity and amylin copper-mediated toxicity. However, we find quite opposite, where batracopene on its own did not have any significant effect on amylin toxicity. In the presence of copper, there was a significant enhancement of amylin copper toxicity of amylin copper complex in the presence of batracopene, which is copper 1 chelator. And this effect of batracopene was not related to its pro-oxidant or antioxidant activity, because no change in, at least in hydrogen peroxide levels, was detected in the presence and absence of batracopene, suggesting actually that the fact of this complex to enhance amylin toxicity in the presence of copper was not related actually to its redox modulating activity, but rather through internalization of complex and disturbing the copper homeostasis in Rin cells. In summary, main findings, we found that copper showed intrinsic ability to stabilize human amylin, its native non-toxic random conformation, to block amylation and block apoptosis, and to form this low affinity complex with featuring low pro-oxidative activity. Redox agents such as NAC can serve as an effective anti-amylin agent, but only under not under elevated copper scenarios, as NAC and copper chelator agent batracopene paradoxically increase amylin toxicity, and therefore that altered copper and or amylin homeostasis may account for this increased activity of or toxic action of copper amylin batracopene complex. We followed this study with mass spectroscopy study. Due to the time constraints, unfortunately, I will not have enough time to dwell too much on the how mass spec can help us in understanding copper amylin interactions, except to highlight the main findings that we were able to publish in collaboration with Dr. Werthe's group, who is expert in mass spectroscopy. For example, mass spectroscopy is a kind of very cool method to show you where exactly copper and other metals may bind, in what oxidation form it can bind, and many other interesting things. Well, shortly, mass spectral data indicated a binding stoichiometry of 1:1, basically confirming our findings about chemical experiments. Collision-induced MS study suggested that is happening here, right here, not surprising, with this amyloidogenic region, and using mutants, amylin mutants, for histidine, for example, to show a dramatic drop in the MS signal, in which copper is bound to amylin peptide, suggesting that HSSN region, and particularly histidine, are one of the important regulators and coordinators that coordinates binding of copper to amylin peptides. And also our previous studies also showed that copper inhibits amylin fibrillation aggregation. We couldn't monitor effect of copper on oligomerization because our study was not sensitive to that, but definitely mass spectroscopy studies showed that copper intercalates and binds to this beta shield enriched fibrils, obviously my time is out, and eventually simulation studies confirmed that copper may binds to this beta hairpin formation, which is represented here within a red circle, coordinated copper, because it contains this negative binding site and consequently can coordinate copper ions, which is actually in a nice agreement with the study with studies demonstrated only and published actually only a few weeks ago, showing that this beta hairpin structure through which presumably amylin aggregates, where copper interacts and bind to, actually can disrupt this fibrillation pathway, and beta hairpin peptide mimics the inhibitory effect of the copper, and providing actually mechanistical insights how actually copper may disturb amylin aggregation. Basically, copper mimics these small molecular inhibitors by disrupting these beta hairpin interactions that leads to oligomerization, first dimerization, then oligomerization, and finally fibrillation. With this, I would like to thank again for the opportunity to discuss work that we are done on the role of copper in amylin aggregation toxicity. We'd like to thank all the students and postdoc and our collaborators and funding, and would like to thank you for your patience, and we're happy to answer any questions that you may have.
Thank you very much for the very interesting talk, and several very interesting aspects that are still in discussion, and questions for understanding both ROS production as well as chelation and other stuff. I don't see questions yet, but let me start with I see first one. Yes, nice work again by Anup Arun Giri. A nice work. I have two questions. One, do you know if copper affects preformed fibrous structures? Maybe start with this.
Well, what we believe it's going on is that copper is binding to the monomeric state when its amylin is still in the monomeric state, because amylin aggregation is a time-dependent process, progress from a monomeric, predominantly monomeric to oligomeric, and then fibril stage. So we started our experiments with a predominant monomeric state. Very few oligomers or fibrils were there, structural studies suggest. So when we add copper in this monomeric, when amylin is predominantly monomeric states, it effectively inhibits dimerization, oligomerization, and then finally fibrillation.
Yes. The second question is, can copper alleviate oxidative stress independent of amylin aggregation?
That's a very good question. We at the point where we can still distinguish between the two. It could be combination of both. Could be that ability of copper to reduce amylin aggregation, but could be also through non-aggregative ability of copper acting on some maybe enzymes, because copper is actually important cofactor in many redox enzymes. So it's possibility that not related to amylin aggregation, copper can reduce amylin toxicity, maybe by stimulating redox activity of certain enzymes. But again, that's very hypothetical in this moment, and we cannot give a definite answer yet.
Okay, another question by Victor Ching. Do other metal ions such as zinc have similar effects?
We didn't explore specifically zinc. We more or less focus on the copper, and I think probably Rams is better to say more about story between zinc and amylin toxicity. So we'll leave it to him if he can answer that question. I don't want to speculate, because again, we didn't do any studies on the role of zinc, but there is in the literature that zinc actually is important regulators. I think we already heard about that, about the important modulatory role of zinc in regulating amylin aggregation and toxic properties.
The next talk will answer that question.
And
Okay. Yes. I think that we are a bit late. Is there any other questions? I don't see any, but we'll have a chance later on if there will be any. Thank you again, Aleksandar, for the very interesting talk. And I move the torch to Kayla.